Okay, today we're going to talk about DNA, right? And we've heard the word DNA before, right? DNA is the stuff we learned about, all genetic material, all living organisms have, et cetera, et cetera. But now we just need to learn a few key points about it. First, let's start off with DNA stands for. So DNA stands for deoxyribose nucleic acid. That's literally where the DNA come from. DNA. Say DNA is deoxyribose nucleic acid, right? And the one thing that we know, by looking at this, is we should be able to look at deoxyribose and know what kind of molecule he is, right? So deoxyribose, and he's really, really important when we talk about DNA. But deoxyribose, remember, he ends in ose. And any molecule, any compound that ends in ose is a sugar. So we know the sugar in DNA is deoxyribose. All right, let's go over some of the stuff that we hopefully remember from when we talked about DNA before, right? So DNA, maybe let's start with the function. All right, you guys think about it. What is my function of DNA? What's he do? Well, his entire job is to store genetic information. All right, that's DNA's job, is to store genetic information. But here's the thing. This genetic information, he's my blueprint, right? And you guys understand what blueprints are for. Blueprints are for building something. Well, this genetic information, this is my blueprint for proteins. Okay, he's my blueprint for proteins. And if you guys remember from way back in Unit 1, when we talked about proteins function was to build a physical organism. So whenever we see like prokaryotes and eukaryotes, and we see different animals and plants and bacteria and fungi, that's because they're made of a bunch of different proteins that were coded for from my DNA. And so whenever we think about DNA is going to give us our proteins, because this stores the information, and then my proteins, that's my physical manifestation of the information. Okay, so there's his function. He stores that stuff. All right, well, let's talk about who has DNA. And hopefully you guys are thinking, it's like, well, what living organisms have DNA? It's all living organisms have DNA, which makes sense, because if I can see this living organism, that means he's made of a physical structure, and then that physical structure was made from the blueprint of my DNA. So all living cells have this. But let's talk about maybe where it's located. Say that's going to differ just a little bit. Where? So when we talk about living organisms, we can actually drop them down into what are my two big cell types for living organisms. Say that's my prokaryote and my eukaryotes. All right, so let's remember pro are my bacteria, and eukaryotes, these are my animals, my plants, my fungi. All right, so let's think about the big difference between them. All right, let's talk about pro. Remember pro means no, so they don't have a nucleus. Versus you do, so they do have a nucleus. And this is actually where my genetic information is kept. So eukaryotes, my DNA is kept inside the nucleus. But prokaryotes, they don't have a nucleus, so I can't keep it in there. I keep my DNA in a nucleoid nucleoid region, which is just where my nucleus would be if they had one. So really, it's just located in the center of my cytoplasm. All right, so DNA stores genetic information that codes for proteins. All living organisms have it. Just depends on what kind of organism where it's located, either in the cytoplasm or in the nucleus. So now let's talk about structure of my DNA. All right, whenever I ask you guys structure, what's the DNA shaped like? Usually you guys say it looks something like this, right? This is called a double helix. Okay, it's like a twisted ladder. Twisted ladder. You know, if you guys go to like those really, really old houses where they have those spiral staircases, essentially what it looks like. But let's think about what that word means. If it's a double helix, how many strands is it made of? Well, double, that means 2. So DNA is a 2-stranded molecule. And you guys can see that because there's 1 and then there's 2. These 2 strands. All right, let's talk about, because remember way long ago when we did our macromolecules, we talked about polymers and monomers, right? So my polymer is my nucleic acid. So now what you guys need to remember is, what is the monomer of nucleic acids? It sounds really, really familiar. The monomer is a nucleotide, okay? So my monomer of a nucleic acid is a nucleotide. And my nucleotides, they're made up of 3 pieces, okay? My first piece is a phosphate group. Remember we talked about phosphates with our ATP molecule. Remember phosphates have a phosphorus surrounded by 4 oxygens, phosphate group. All right, my next piece in a nucleotide is a sugar. And we already talked about what sugar my DNA has, because that's literally what the D stands for. So my sugar is deoxyribose. And then my last piece of a nucleotide is my nitrogen base. So if I look at these 3 things, I can actually tell you what elements I find in a nucleotide. Well, if I look at phosphate group, I know it has phosphorus and oxygen. Sugars, if you remember, are made up of carbon, hydrogen, and oxygen, but I already have it listed there. And then my third one, nitrogen base. Well, that looks kind of weird. So remember, we always said our elements for a nucleic acid are punch. All right, now if I'm looking at this nucleotide, these 2 things right here, my phosphate group and my sugar, this is what's usually referred to as the backbone of my DNA, which means it's on the very, very outer side. It's really, really sturdy. And then my nitrogen base, because we're going to make it kind of look like a ladder here in a second, these are my rungs. And you guys know, if we've drawn, or if you've all seen a ladder before, so here's my ladder, and then here's the rungs in the middle. Well, my phosphate groups and my sugars, those are the outside, and then the nitrogen bases, those are the rungs in the middle. Now my nitrogen bases, I actually have a couple different options for those, right? I have 4 different nitrogen bases. I have A's, because we usually just use letters, but I'll tell you what the words mean. I have A. A means adenine. T means thymine. C means cytosine. And then G is guanine, right? Now these nitrogen bases, what's really, really cool about them is they actually pair up in a very specific way, right? This guy named Chargaff, he's the one that discovered it, so we call him Chargaff's rules. Now A's always pair with T's, and C's always pair with G's. An easy way to remember it are apples grow on trees. Oops, that looks kind of like an I now. Apples grow on trees, and cars go in the garage, okay? Apples on T's, cars in garage, and we'll use that here in a little bit. But really what's cool about these nitrogen bases, it's that it's the sequence, and we'll talk way more about this later, but it's the sequence of these that give us our differences. Because every living thing on this pretty little green earth of ours are made of the same 4 letters. So like your 4 letters are the same as my 4 letters, but I have freckles and maybe you don't. That's because of the order of our nitrogen bases. Or if you guys are looking at like a fish or something in your classroom, well, those fish are made of the same 4 letters, but because they're in a different order, that gives us a different organism. Or if you look at the plants, you know, in the classroom or outside, it's the same 4 letters, but because they're in a different order, that gives us different things, which is really just kind of crazy, but we're going to do more with that here after a while. So what we want to do is we want to draw our nucleotide. All right, now we said phosphate was first. So here's my phosphate group. We're just going to make it a circle. And then he's going to attach to my sugar. We'll make that a triangle. And then sugar will attach to our nitrogen base. Okay, so this right here is a nucleotide, a phosphate, a sugar, and nitrogen base. But remember, nucleic acids are a polymer, and they're made of monomers. And if we remember correctly, monomer, this is one thing. So I have to put a bunch of these things together. I have to repeat this. So I have to repeat the monomer to get a polymer. And so now we can draw that. So let's maybe make it in a different color. There you go. All right, so let's start. I have a phosphate, a sugar, and then a nitrogen base. And then I just repeat it. Phosphate, sugar, nitrogen base. And let's do one more. Phosphate, sugar, nitrogen base. But here's the deal. When we talked about DNA structure, we said he's a double helix. So how many strands do I have to have? I have to have 2, so I just have to double it on the other side. So here's my nitrogen bases. Whoops. So I'll put a nitrogen base in the middle. Attach that to a sugar. And then a phosphate group. And I'll do the same thing. Nitrogen base. Attach to a sugar. Attach to a phosphate group. Nitrogen base. Sugar. Phosphate group. So now I've made 2 strands, right? Because this would be strand 1. This would be strand 2. I have a double-stranded molecule. But I don't want this to fall apart, so I'm actually going to have bonds right here in the middle that hold these nitrogen bases together. These are the weakest bonds in biology. Do you guys remember? What were my weakest bonds in biology? That's a hydrogen bond. So hydrogen bonds hold my DNA together, okay? And we'll talk about why we want to hold them by really, really weak bonds here in a second. But if I look, so this right here, this is one nucleotide. And so I just keep repeating those nucleotides to get a nucleic acid. And then if I look, this phosphate in this sugar, we'll do this one, this phosphate in the sugar, this is my backbone because it's the very, very outside. And then these nitrogen bases, these are the rungs of my ladder because they're in the middle, and that's why I drew that nice little ladder like this. So I have the phosphates and the sugars on the outside, and then I have the nitrogen bases in the middle. And I have to do that because DNA is 2 strands. But what I want to do next is I want to make a copy of my DNA because maybe I need to make a new cell, right? If I need to make a new cell, that means I need to make more DNA so each cell has the right amount. So what I'm going to do next is I'm going to make a new copy. And I'm going to do it by a special process by doing that's an R, replication. Okay, so replication, right? Replication, just to be redundant, this means I'm going to make a new copy of DNA, right? And the reason I want to do that, you guys don't have to draw this, but just kind of understand. Like, let's say here's my cell, and I'm going to split this cell into 2 cells, so that way I can make more, right? And maybe I had a stick of DNA. Well, if I don't double my stick of DNA, one cell would get DNA, and the other cell would get nothing. Well, would this cell right here, would this work properly if it doesn't have DNA? No, it won't. But if I double this DNA, so if I have 2 yellow sticks before I divide, then each cell would get 1 copy of my DNA. So that means it would be functional. So I always have to, and if I kind of draw it like this, oops, there you go. So here's my cell. So he had 1 stick. I doubled it, and then I split into 2. So now this cell that I started with and the cells I end with are exactly the same. Now the first thing I have to do, if I want to make a copy of DNA, is I have to break those hydrogen bonds. So that's my very, very first step, is I need to break the hydrogen bonds. Because right now those hydrogen bonds are holding that DNA together. So if I draw, let's see here, if I draw a pretty DNA, let's make this pretty blue color. So here's 1 strand of DNA. And let's just make up a sequence. Maybe it goes A, T, T, C, G, T, A. There you go, it's 1 sequence. Well, remember, DNA is double-stranded, and so I have to make the other strand of it, and I used to use Chargaff rules. So A's, remember, apples grow on trees, so T's go together. And what do trees have? Trees have apples, trees have apples. C's, remember, C's go with G's because cars go in the garage. And what do garages hold? They hold cars. Trees have apples, apples have trees. And so there, and then we'll do our hydrogen bonds. So there's our starting molecule, right? This is our starting DNA strand. Now what I'm going to do first, remember first I have to break the hydrogen bonds. So let's just have this little triangle. So this triangle is going to go up, and he's going to start breaking all of those hydrogen bonds. Because when he breaks all of those hydrogen bonds, that's going to separate my DNA. So now my 2 strands are apart. So let's go ahead and write our letters again. So this went A, T, T, C, G, T, A. And then we have the opposite of it. So T, A, G, C, A, T. There you go. So I broke all the hydrogen bonds, and then they got split apart. But now I can use Chargaff rules again, and I can make a new strand. So if I start here with this first strand, because there's strand 1, there's strand 2. See, there's strand 1, there's strand 2. So I'm just going to use my Chargaff rules. So A's, apples, go on trees. Trees grow apples. Trees grow apples. Cars go in the garage. Garages hold cars. Trees have apples. Apples have trees. And I can do the same thing to this side. So trees hold apples. Apples, trees. Apples, trees. Garages, cars. Cars, garage. Apples, trees. Trees, apples. And then I'll make my hydrogen bonds here in the middle. So see, look, I started off with my original DNA right here. But then when I broke those hydrogen bonds apart, now how many strands do I have? I have 2 strands, because I have this strand right here, I have this strand right here, which means when I do, because this was replication right here, when I do replication, in the end, I get two identical strands. 2 identical strands, right? Now there's actually a really fancy process that we just did. It's actually called a semi-conservative process. Well, let's spell it right. Conservative. This is a semi-conservative process. And if you think about what those words mean, right? Semi means sometimes, and if I conserve something, I save it. So I used half my old strand to make a new strand. Because remember, if I look over here, my original strand, it was made up of 2 blue strands. But then when I split them apart, I used the blue strand to help make the new part, the yellow strand. So that's why it's a semi-conservative process, because I used old parts to make a new part. All right, let's recap here. So DNA stands for deoxyribose nucleic acid. Deoxyribose is a sugar because it ends in ose. My function of DNA is to store genetic information, which will build my physical organism. So it goes from DNA to proteins, holds my information, physical manifestation of it. All living things have DNA, just depends on where. If I'm a prokaryote, it's going to be in the cytoplasm. If I'm a eukaryote, it's in the nucleus. DNA is made of a double helix, which means he's 2 strands. And those 2 strands are just a bunch of repeating nucleotides, because that's my monomer. My nucleotide is made up of 3 things: a phosphate, a sugar, a nitrogen base. The sugar is deoxyribose, because that's where the D comes from. And then my nitrogen bases are the A's, the T's, the C's, the G's. And they go by those Chargaff rules. So here's my nucleotide. Remember this right here, this is my backbone. And then these nitrogen bases, these are going to be the rungs, or the middle of it. And so I just have to repeat those over and over and over again. So this was a nucleotide, this is 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 a nucleotide, because it's just repeating of a phosphate, a sugar, a nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Same on this side. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. And then those 2 strands, so that makes up the 2 strands for the double helix. So here's strand 1, here's strand 2. They're held together by those hydrogen bonds, which are really weak. The reason I want them to be really weak is because when I do replication, which is when I make a new copy of DNA, I can break them apart. So that way I can use the old strand as a template to make my new strands, and then they're identical. All right, I hope that helps.